Welcome back to the deep dive. You know, our mission today is it's pretty fundamental. We want to stop guessing and start really understanding how we build muscle,
Exactly.
whether that's for, marathon level endurance or just raw explosive power.
That's the goal. And the sources we're diving into today are, they're pretty dense. We're going from what you can see, the muscle itself, right down to the protein level changes that dictate everything.
So we're talking about the structure, right?
Yeah.
Fast twitch versus slow twitch.
All of it. The structure, the fiber differences, and the energy systems, aerobic versus anaerobic. And the most important part is connecting specific training techniques to the molecular and even the epigenetic shifts they cause inside your body.
I love that. Connecting the gym floor to the genome.
Yeah.
For too long, we've just talked about muscle as this bulk thing.
Right.
But today we're treating it like the sophisticated engine it actually is. Okay, so let's unpack this. Let's start with the literal nuts and bolts,
Okay.
the architecture of movement.
We have to start with structure because structure defines function. When you look at skeletal muscle, the voluntary stuff we use to move, it's incredibly organized.
It's built in layers.
Layers, exactly. You have these bundles called fascicles, and they're made of myofibers. A myofiber is basically just a giant muscle cell.
So the myofiber is the core building block.
Yes, and if you go inside those cells, you find myofibrils, which are these long chains of repeating units. And the smallest, most fundamental unit in that chain, the one that actually generates force,
That's the sarcomere.
that's the sarcomere. You can think of it as the microscopic piston in your muscle's engine. And of course, that whole engine is packed with blood vessels and nerves for fuel and communication.
That molecular piston analogy is perfect.
Mhm.
So how does that piston actually fire? I mean, it's not just two ropes pulling, right? It's a whole choreographed sequence.
It is. It's called the cross-bridge cycle.
Yeah.
And the ignition key, the thing that starts it all.
Calcium.
Calcium. When a nerve impulse hits, calcium just floods into the muscle cell's interior. It's the ultimate chemical switch. This free calcium goes looking for a very specific protein on the actin filaments called troponin C.
And troponin C is really sensitive, isn't it? It can bind 4 calcium ions, and it does so cooperatively.
Precisely. So a small release of calcium triggers a huge response. Once those 4 sites are full, troponin C literally changes its shape. It yanks its partner protein tropomyosin out of the way.
And tropomyosin is like a safety lock.
It's the safety lock. When it moves, it uncovers the binding sites on the actin filament and the myosin heads. Well, they've been waiting to grab on.
So they swing out, they form that cross bridge.
Mm.
What's next? How does the actual shortening happen?
This is where the energy comes in. So the myosin head is stuck in this stiff 45-degree state. We call it the rigor state. It needs to be reset.
And ATP does that.
Right. An ADT molecule binds to the myosin head, and that binding alone makes the myosin briefly let go of the actin. It breaks the cross bridge.
Which is what stops your muscles from just locking up solid.
Exactly. Then the myosin splits that ATP into ADP and a phosphate ion. That energy release, it cocks the myosin head back like a trigger, allowing it to weakly reattach to the actin, but now at a wider 90-degree angle. It's loaded.
And the power stroke, the pull.
That happens when the phosphate is released. That's the trigger. The myosin head snaps forward, it rotates forcefully, and it pulls the actin filament past it. That's what shortens the sarcomere. That's force. Then the ADP leaves, and it's back in the rigor state, waiting for the next ATP.
A rapid ATP-fueled ratchet mechanism. It's incredible. Okay, so that explains how a muscle contracts, but it doesn't explain the difference between, say, a marathoner and a powerlifter. If they're both using the same basic engine, how are their capabilities so different?
And that brings us right to the muscle fiber divide, speed versus stamina. This is where that molecular specificity just shines.
Because skeletal muscle isn't all one thing.
Not at all. It's heterogeneous. You have fibers with fundamentally different blueprints, different speeds, different energy demands.
Let's start with the endurance specialists, the type I, or slow twitch fibers.
Right. These are built for the long haul. They express a protein called MYH7, that's myosin heavy chain 7, and that dictates a slow contraction speed. But they are incredibly fatigue resistant.
Because their whole energy system is different?
It's built for sustained output. They rely almost entirely on oxidative phosphorylation, or XPHOS. So they are just packed with mitochondria, and this is important. They have a really fast mitochondrial turnover rate. They keep their power plants clean and efficient.
Okay, then on the other side you have the sprinters, the powerlifters, the type 2 fast twitch fibers.
Type 2 fibers contract quickly, explosively, but they tire out fast because they're running on a different system, mainly glycolysis for quick, short-term energy. We even subdivide them based on their protein. Type Cia expresses MYH2, and the fastest ones, type 10, express MYH1.
So the difference between a sprinter and a marathoner is literally which protein their muscle cells are told to build.
At a fundamental level, yes, that's the molecular specificity we're talking about.
And what's really wild is that this profile isn't set in stone. Muscle is. It's plastic. It remodels based on what you ask it to do.
It's highly plastic, but that's a double-edged sword, right? Especially when you're injured or inactive.
How so?
Well, in studies looking at disuse atrophy, think about having your arm in a cast. The fast-twitch fibers, the ones for explosive power, they showed a much, much greater reduction in size compared to the slow-twitch fibers.
So the explosive type 2 power that you work so hard to build is actually the first thing to go when you stop training.
It's the most vulnerable. It wastes away faster. It's a critical insight for anyone recovering from an injury. You have to have a strategy to preserve those fast-twitch fibers because they will suffer the most.
Okay, so let's talk fuel. We have these two different engines, fast twitch and slow twitch, and they both need ATP. How does the body make ATP for a 30-second sprint versus a 2-hour run?
ATP is the currency, right? And muscle has two major ways to make it. For quick, immediate power, you use anaerobic glycolysis. This happens outside the mitochondria in the cytoplasm.
Right. It takes glucose, breaks it down into pyruvate, and you get a small but very fast burst of ATP.
Correct. And in those moments of maximum effort when energy demand outpaces oxygen supply, the very last step is converting that pyruvate into lactate. That's your system for a 30-second all-out burst.
And then there's the other pathway, the type I endurance fibers love.
That's aerobic oxidation, OXPHOS. This happens inside the mitochondria. That pyruvate gets converted to acetyl-CoA, it fuels the TCA cycle, and that powers the electron transport chain. You get way more ATP this way.
A huge amount more.
But it requires a steady supply of oxygen. It's the system for running the long game.
You mentioned lactate. For decades, anyone who worked out was told that lactate was it was toxic waste, the thing that made your muscles burn.
Right.
But the science on that is completely flipped.
It's been a total revaluation. That old view is it's dangerously outdated. Lactate isn't metabolic garbage. We now know it's more like a lacthormone. It has signaling properties. It's a fuel. Absolutely. It can be immediately oxidized for energy inside the mitochondria. And this leads to the lactate shuttle concept.
Which changes everything about how we think about energy distribution in the body.
It really does. The shuttle explains how lactate moves from, say, the driver cells, your fast-twitch fibers, making a lot of it, to consumer cells. That includes your slow-twitch fibers, your heart, and even your brain. They use it directly as an energy source. It's fuel on the move.
This is where it gets really, really interesting for me.
Right.
If we know the structure, the fiber types, and the energy systems, now we can talk about how training actually forces these changes.
Exactly.
So let's start with endurance. If I go for a long run, how does that molecularly push my fast-twitch fibers to become more like slow-twitch fibers?
Endurance exercise is a primary driver for that transition, and the mechanism is all about increasing the density and the health of your mitochondria. We call it mitochondrial biogenesis.
And there has to be a signal that tells the cell nucleus, hey, we need more mitochondria.
There is. Endurance work generates molecular stress, and that activates something called the ROS-AMPK pathway. You can think of AMPK as the cell's energy sensor.
Like a low-fuel light on a car's dashboard.
That's a great way to put it. When your ATP levels drop, AMPK senses it and gets activated, and its activation is the crucial first step.
So what does that low-fuel light trigger?
It triggers the master regulator, PGC1 alpha. PGC1 alpha is like the general contractor for building new mitochondria. So you stress your energy systems, AMPK activates PGC1 alpha, and your body gets the signal to build more powerhouses.
And it goes even deeper than that. The sources we looked at pointed to actual epigenetic changes to how the DNA itself is packaged.
It's incredibly specific. The core mechanism involves regulating histone methylation. Think of that as like a dimmer switch on your genes. Exercise can induce that shift to slow twitch by literally flipping the switches on the genes for mitochondria and the myosin heavy chain isoforms.
So it's not just turning genes on, it's making them easier to access.
Yes. For example, endurance exercise reduces one type of methylation and increases another right at the promoter for the MYH7 gene. That's the slow twitch gene.
It's basically unlocking and highlighting the instruction manual for building slow twitch fibers.
That's a perfect analogy. It is the ultimate proof that training is direct communication with your genes.
Okay, that's endurance. Let's switch to strength and speed. We saw a fascinating study comparing two types of resistance training, slow speed resistance training, or SRT,
Right, like a 3-second down, 3-second up tempo.
versus traditional resistance training, TRT. And they both improved strength, but the adaptations were very different.
They were. They were specific and in some ways non-overlapping.
And here's the part that blew my mind. We're always told speed builds speed. But this study said the slow, lower intensity SRT actually produced better sprint results.
It did. The SRT group, which was only using about 50% of their 1-rep max, showed much greater improvements in rapid force contraction capacity. And they had significantly better 30-meter sprint times than the traditional heavy lifting group.
That is genuinely shocking. How is that possible?
Well, the leading hypothesis is about blood flow. That sustained, slow contraction likely restricts blood flow to the muscle, creating a low oxygen or hypoxic environment.
So that hypoxia might trigger a different set of adaptations.
It might trigger specific pathways that actually enhance the contractile elements needed for rapid movement, even though the training itself was slow. The TRT group did get better at what they trained for, producing maximal force at slower speeds. But the big takeaway is that the speed of execution creates unique adaptations, independent of the weight.
This is all incredible for performance, but it also has huge implications for just general health.
Yeah.
Let's connect these training modes back to managing chronic inflammation, which is a major concern, especially as we age.
It's a critical application, and the data is clear. Aerobic, resistance, combined training, HIIT, they all significantly reduce key inflammatory markers like IL-6, TNF-alpha, and CRP. Exercise is fundamentally anti-inflammatory.
Was one mode better than the others?
It depended on the marker, which really reinforces why you need a mix of training. Combined aerobic and resistance training was the best for lowering IL-6.
Okay.
HIIT was best for reducing TNF-alpha, and resistance training on its own was particularly good for improving CRP levels. So for you, the key is that all movement fights inflammation, but to get the best systemic effect, you need to diversify your training.
So if we tie all of this together, what's the big picture?
The big picture is that there's this profound molecular control behind everything. Muscle plasticity, metabolism, training success. It all comes down to these pathways. Whether you want endurance or power, the key is selectively activating things like the AMPK, PGC1 alpha axis through very precise training.
So what does this all mean? We've seen that your contraction speed, your fatigue resistance, the health of your mitochondria, it's all fundamentally linked. And training is essentially just you speaking to your genes.
You're telling them exactly what machinery to build.
Exactly. And just to leave you with a final provocative thought, consider how far this science is going.
Yeah, this is pretty mind-blowing stuff.
Researchers are now seriously exploring mitochondrial transfer. I mean, injecting healthy mitochondria into damaged cells to restore function.
For diseases like Alzheimer's, Parkinson's, heart damage from ischemia.
Right. So if scientists can literally transplant these tiny energy factories to fix broken tissues,
Yeah.
what does that say about the power you have right now with exercise, the power to optimize and build up your own internal network for lifelong health, something profound to chew on during your next workout?